Hello everyone and welcome back to Willow's Notes. In our last video, we talked about the Operon. The Operon was an example of a repressible operon. Today, we will talk about an inducible operon, which means an operon that is not active and is induced to become active. The classical example of an inducible operon is the lac operon. Lac for lactose. Let's briefly remember the structure of lactose together. Lactose is a disaccharide, meaning it is a sugar composed of two units, galactose and glucose. Once ingested, it needs to break down. This bond needs to break down, and the enzyme responsible in this hydrolysis is beta-galactosidase. The gene for beta-galactosidase, LAC-Z, is located in the lac operon. Just like the operon, the lac operon has its promoter and operator. Also, similar to lac has its regulatory gene upstream called LAC-I. LAC-I codes for a repressor protein. Do you remember the repressor of the operon? It was inactive, right? Meaning it didn't have any affinity to the operator. While here, this repressor is active. And when there's no lactose in the environment, why waste energy and resources in synthesizing an enzyme that's not needed? Therefore, the operator needs to be blocked, right? So this active repressor binds to the operator, switching it off. When lactose becomes available again, for example, if a human host drank milk, lactose will be available to the E. coli in the colon. Allolactose is an isomer of lactose. It forms when the bacteria take in the lactose. Allolactose binds to the repressor. Remember, just like in the operon, this repressor is an allosteric protein, which means the binding of the allolactose to it changes its shape, and it can no longer bind to the operator. In this case, allolactose is the inducer because it induced the change. It unblocked, let's say, the operator, allowing RNA polymerase to proceed in the transcription of the LAC-Z gene. And this was it for today's video. I hope you found it useful and easy, and I'll see you in the next one. Bye!